So tonight's video is going to focus on cell cycle and then regulation of that cell cycle and how those cells are able to control when the cell is dividing and how often. So we're going to start with the cell cycle and the parts of the cell cycle. So the cell cycle itself is a regulated series of events that allows the cell to grow and to reproduce. So some key attributes here of the cell cycle is that it's regulated. It is controlled. It doesn't happen whenever the cell doesn't move through the cell cycle whenever it feels like it. Okay? And the purpose of the cell cycle is for the organism as a whole to grow. So basically to get larger. And so the cell itself will grow, and then the cells will reproduce, which will allow for the organism to get larger. So the cell cycle itself has basically three parts. It has interphase. So interphase, if we're looking at our diagram over here on the right, interphase is the bulk of the cell cycle. You'll notice on the smaller inner circle here, interphase is this whole area here. So this should all look familiar from last year. So interphase is the main part, is where the cell's going to spend the most of its time, and we're going to go step by step into each one of these different parts of the cell cycle. There's mitosis as well, which is the actual division of the nucleus. So not the division of the cell itself, but technically mitosis is just the division of the nucleus. And then the little bit of cytokinesis, which is the actual division of the cytoplasm, so the cell actually splitting into two. And again, we're going to go through each one of these individually. You'll also notice on this diagram, we have what's called G naught over here. It looks like G0. It's pronounced G naught. So in G naught, the cell is no longer dividing. And so cells that enter G naught, they're not actively participating in the cell cycle. So they are not going to be actively dividing at any point. They can re-enter the cell cycle if they get certain environmental cues or information from other cells. But as a general rule, they are staying out there in G naught. So they're not getting a lot larger because they're not preparing for division. They're not replicating their DNA. They're just hanging out, doing their thing. Non-dividing cells can also exit the cell cycle or just be kind of held up at any particular spot. So maybe they're held up during G1. Okay? So non-dividing cells can also go ahead and exit at any point, as well as get kind of held up, maybe slowed down by a checkpoint or something. All right, so let's look at interphase. So interphase is divided into three parts. And again, this is where the cell spends the bulk of its time. If you look at your diagram over here, more than 3/4 of this circle, right, is spent in interphase. So about 75%, 80% of the cell's life is spent in this interphase. It's not constantly dividing. It's constantly doing its normal things. So the first kind of phase or step in interphase is G1, which is considered what's called our first gap phase. This is a growth phase. The cell is doing its normal growth here. It's producing organelles. It's increasing what's in the cytoplasm. It's active. You notice right here it says metabolically active. So that means the cell is functioning. It's functioning, it's working, it's doing whatever its normal job is, as well as continuing to grow, to increase what's in it, the organelles that are in its cytoplasm. It's starting to prepare for eventually doing cell division. The next part of interphase is that S phase. S is for synthesis, right? Remember, synthesis means that we're making something new. So in this S phase, we are replicating that DNA. That's what's being made. That's our new thing that we're producing during the DNA, during the S phase. So the cell is continuing to grow, and during this S phase, it will copy its DNA, and it will make an exact copy of its DNA. So it will double the amount of chromosomes that are in that cell at that given point in time. Because as the cell divides, it's going to need to have extra chromosomes, enough chromosomes to split between two cells. These chromosomes are replicated, and they form sister chromatids. If you were in my class, we most likely did the chromosome dance when you were a freshman. So it's forming these sister chromatids that are going to be held together at the centromeres. Remember, your sister chromatids were your arms and your legs. Your centromere was your belly. and they were held together. So that's our replicated DNA there. So the cell, though, is also, in addition to making a copy of its DNA, it is still continuing to grow. So you will, when the cell exits this S phase, it will have two identical copies of its DNA so that it can effectively split that DNA out into the new daughter cells. The last part of interphase is our second gap phase. So our second gap phase, the cell is again continuing to grow. So that G, sometimes you'll see diagrams where that G is represented as growth, not gap. So in that second gap phase, the cell is continuing to grow and it's getting ready to do cell division. A lot of protein synthesis is happening. A lot of enzymes are being made because these are all things that are going to help regulate and control cell division. We've got some ATP being produced so that there's energy for this to happen. Centrioles are being replicated. Centrioles are important for helping the chromosomes split apart and move to the opposite ends of the cell. So there's a lot of things going on in the cell right now as it's getting ready to divide. So after interphase, the cell is going to move into mitosis. Mitosis is important for an organism, for both multicellular and unicellular organisms. It's needed for growth. It's needed for repair, right? If I have an injury to my skin, I want to replace that with identical skin cells. So I want to make exact replicas of the cells that were damaged. So it's going to help repair organisms. Some unicellular organisms will use it for asexual reproduction, okay, where they now went from one to two organisms. Okay. The mitosis part of the cell cycle is going to alternate with interphase. So the cell cycle is constantly happening, right? That circle is constantly going, and so the cell is constantly moving between interphase and mitosis, interphase and mitosis, right? It divides, those cells go through interphase, they are ready to divide, then their daughter cells go through interphase. Okay? And this, again, is technically the division of the nucleus. The splitting of the cytoplasm is that cytokinesis. So if y'all think back to your freshman year, remember you had 4 steps to mitosis. So the cell cycle as a whole includes interphase, mitosis, and cytokinesis. These phases here are just part of mitosis. So they're just a tiny little part of this cell cycle. So remember your PMAT acronym to help you remember the order that these things are happening in. Prophase, metaphase, anaphase, telophase. Again, some reminders of what happens during each one of these. Again, these are things hopefully from your freshman year that you remember or will maybe trigger some memories here. So in the first step, in prophase, I need the nuclear membrane to start to break down. I need to be able to release the DNA, right? And again, this is division of the nucleus. So in prophase, right here, my nuclear membrane is starting to break down. The chromosomes, they're going to start to, the chromatin is going to start to condense down into chromosomes. So you're going to start to see those chromosomes. If you look at this first one here, where the cell is an interphase, it's hard to delineate the individual chromosomes. So as prophase is occurring, that chromatin, that loose DNA there, will condense into these chromosomes. And you can see in the prophase how the DNA looks different, how it looks more like these individual strands. You can see it a little bit better. So I've got, during prophase here, I've got the nuclear envelope breaking down. This chromatin is condensing into chromosomes. And I'm starting to see these spindle fibers. Okay, you can see there the yellow in the diagram here, but you can see these spindle fibers are starting to get laid down here. So the centrioles, okay, I'll spell that for you too. So here's the centrioles, they're going to start laying down those spindle fibers. So all of these things are happening to the cell when it's going through prophase. So right as it's entering this mitosis part of the cell cycle. So from prophase, we're not going to concern ourselves with prometaphase. Okay? It's just kind of a weird kind of limbo-y place that the cell is in between prophase and metaphase. So from prophase, the cell will move into metaphase. So at metaphase, at this point, the centrosome, so let me erase some of this so you can So the centrosomes, so our vocabulary is going to be really important as we move through this unit because it's all basically going to sound the same. Okay, so the centrosomes are going to be at opposite ends of the cell. So that's where the centrioles basically have migrated to the end there. So the centrosomes, where these spindle fibers are laid down, are going to be at opposite ends of the cells. And the chromosomes are lining up right here in the middle. So that's my condensed DNA in the form of chromosomes. So they're lining up on what we call the metaphase plate. They're lining up along the equator of the cell. Remember, this is still replicated DNA. So I still have twice as much DNA as this cell needs. So from metaphase, so once this cell has lined up the chromosomes along the equator and moved the centrosomes to the end so that these spindle fibers are ready, the cell will shift into anaphase. And during anaphase, those chromosomes are going to get separated. They're actually going to get the sister chromatids. So one more C word we're going to throw in there. Okay, so these sister chromatids are going to get separated. Those sister chromatids, those are those replicated chromosomes. I can't spell. So those sister chromatids are those replicated chromosomes. And so we're going to split those up. I don't need multiple copies. I need a copy of everything in both cells. So I need those duplicates to split apart. So during anaphase, you can see they look like little V's here separating out. These sister chromatids are going to split apart. So if you remember, when this chromosome is replicated, again, thinking back to freshman year, right? I've got this chromosome, kind of looks like a big X. The centromere is right there in the middle, right, holding them together. And so these two on the bottom would be sister chromatids. These two on the top would be sister chromatids. So when I go through anaphase, I can pull these guys away one direction and pull these guys away the other direction. So I separate those sister chromatids, which are the ones on the top and the ones on the bottom. So I pull them apart there at that centromere. So the centromere, again, I'll give you the spelling. The centromere is that central part holding them together. So in anaphase, those sister chromatids are going to separate. They're going to basically get pulled by those spindle fibers towards the opposite ends of the cell. And so that'll lead us into telophase. So in telophase, basically, I need the opposite of prophase to happen. I need the nuclear membrane to come back. So I need to reform the nuclear membrane around because I need to trap that DNA in a nucleus again. Okay? The chromosomes will unwind back into chromatin, and those spindle fibers will start to break down because I don't need them anymore. So again, I've got the opposite of prophase happening here. Okay? I've got the nuclear membrane reforming. I've got the chromosomes unwinding back into chromatin. And then I have the spindle fibers breaking down because I no longer need them. And during telophase, this is when I'm also going to start to see cytokinesis start. So our cytokinesis is going to be the actual division of the cytoplasm because we just divided the nucleus up during the PMAT, during the mitosis part. So it actually will start in a little bit in very late anaphase or the very beginning of telophase. And it's going to look a little bit different depending upon the type of cell that it is. If it's an animal cell, because there is no cell wall, right, I'm going to see what we call a cleavage furrow. Basically, I get a belt of proteins that will form around the middle of the cell, and it will just tighten, and it will squeeze together. That's where I get this cleavage furrow here, until eventually it pinches the cell into two different cells. A plant cell can't do that, though, because a plant cell has a cell wall. So a plant cell can't squeeze together its cytoplasm until they pinch together fully. So a plant cell will lay down what is called a cell plate. And you can see that laying down right here. Basically, it starts building the wall between the two cells. And that wall, that template for that wall is that cell plate. And so once that cell plate is completely built and fused into the cell membrane, it will eventually divide the cell by a wall into two different parts. So again, the basis of cytokinesis is the same. I'm dividing the cytoplasm up so that I end up with two separate cells, but it will look a little bit different in my different types of cell, animal versus plant cell in particular. So let's start looking at how this is regulated. I feel like I kind of ran through that really fast, but hopefully you realize that most of that is review from your freshman year. Stop, rewind, listen to it again, but I don't want to keep repeating it because it will just slow us down. Okay, so let's shift over to what it looks like when we try to regulate this and we're controlling this. So the cell is going to use checkpoints to help regulate the cell cycle. Those checkpoints are going to use those transduction pathways that we've talked about. So those pathways that, amplify the signal and pass the signal along. Okay? And so these checkpoints are going to help regulate the cell cycle. They're going to give it both stop signals as well as go signals. It will give both of those. Okay? If everything looks good, it will tell it to go ahead and go through division. If something doesn't look right, it will tell it to stop and don't do that. So here's where we get into the stuff that is new. That we looked at checkpoints your freshman year, but we didn't really look at kind of how this stuff is working. So these checkpoints are going to be controlled by what we call cyclin-dependent kinases. Okay, remember a kinase from our transduction pathways. These protein kinases, these are enzymes, right? ACE, ASE. These are enzymes that activate or inactivate proteins, and they're going to do that by phosphorylating them, by adding phosphates to these proteins. So if you look at this diagram to the right here, you can see that phosphorylation is happening, dephosphorylation, right? I've got active forms versus inactive forms, and that's all coming because these protein kinases are phosphorylating these molecules, and that will turn them on and turn them off. And so these protein kinases, these cyclin-dependent kinases, their concentration remains the same in the cell. They stay constant. We talked about this when we were talking about how it was beneficial sometimes to a cell to have parts that can be turned on and off because it means it could happen really fast. We don't have to wait to create it. And this is one of these cases. So these cyclin-dependent kinases, which are abbreviated, you'll see these a lot of times, just listed as CDKs. Okay, so the CDKs, their concentration remains constant, and they get turned on by another chemical called cyclin. And the cyclin's concentration is going to fluctuate within the cell. And that's why it's called cyclin. It goes through cycles of being a lot and being a little and being a lot and being a little. And so these protein-dependent kinases, or these cyclin-dependent kinases, these protein kinases, they depend upon the concentration of cyclin to either turn them on or turn them off. So they're there all the time, and they just get turned off. The cyclin is the thing that needs to build up, and then it drops down again, and builds up and drops down again. Okay, so we see their activity, the activity of the CDKs, that's going to change based upon the cyclin concentration, but the concentration of CDKs stays the same. It's just what they're doing. So we're going to look at something in particular, okay, what's called an MPF complex. So the MPF complex stands for maturation, right? Like I'm becoming mature. So maturation, and I cannot spell tonight. Maturation, that's an R. Promoting factor. So MPF encourages or promotes the cell to mature, okay, to go through maturation. Okay, MPF is also sometimes called the cyclin CDK complex. Okay, again, MPF or maturation promoting factor is also sometimes referred to as the cyclin CDK complex. Because again, remember those CDKs are dependent upon cyclin to activate them. So MPF is going to increase or rise to its peak during the synthesis and G2 phases of interphase. Okay, and it will fall really quickly during mitosis. Okay, and so you can see on this graph over here on the left, you can see these various cyclin concentrations going up and going down. and changing in concentration depending upon the part of the cell cycle that we're in. So an increase of concentration in MPF is going to push the cell past the G2 checkpoint. And so the G2 checkpoint, which would happen right in here, okay, is making sure that the cell is ready to go through mitosis. In particular, at that G2 checkpoint, the cell is checking the DNA. Was it copied properly? Does it have any problems? Because I don't want to go ahead and divide and split that DNA out into new cells if that DNA is faulty. And so there a rise in the cyclin at this stage, okay, and then activating those CDKs will push the cell over into mitosis. Okay, and so depending upon the checkpoint that the cell is trying to get past, I'm going to have different cyclins that will push them past. Okay, again, if you look at the diagram, you can see M cyclin, S cyclin, G1, S cyclin. So those cyclin concentrations, every time they increase, that's because they're trying to push the cell past a checkpoint. Okay, and if the cell isn't meeting certain criteria, then those cyclins won't increase, and so it won't push the cell past the checkpoint. Like, let's look at some of these checkpoints and see what they would be looking for. So the first checkpoint we're going to look at here is the G1 checkpoint. So the G1 checkpoint is a restriction point in mammals. So your cells are going to go through this checkpoint here. Okay, once the cell gets through G1, it irreversibly commits to cell division. Meaning if the cell gets through G1, it's not leaving for G0. It is going to keep powering through until it gets to mitosis, until it gets to cell division. So it irreversibly will commit now to cell division. It will not pop out into G0 if it passes this point. Okay? At this checkpoint, your cell is looking to make sure that DNA and other factors weren't damaged, so that the cell can have enough energy in its reserves as well to be able to push through to cell division. If it's not ready, then the cell can be sent to G0. Okay? But if it says it's ready and pushes it past here, then the cell can't go to G0. So this checkpoint could force it over into G0 if it thinks it's not ready to keep powering through to mitosis. Maybe something went wrong with the DNA. Maybe it's missing certain other factors that it might need. Maybe it doesn't have enough ATP available. Okay, and so at this G1 checkpoint here, okay, it's either going to head off into G0 or keep committing to cell division. The G2 checkpoint is happening right here before mitosis. Okay, this is the one that is checking for proper DNA replication. Okay, does the cell have enough and accurate DNA to pass on to the two new daughter cells? It's also going to check the size of the cell. Basically, the cell needs to be big enough to be split in half and still have enough resources for the two new cells. It's also going to check its protein reserves. Does it have enough proteins, enzymes in particular, to get it through these next couple of stages during mitosis? The last checkpoint we're going to mention here is the M checkpoint, which is the metaphase checkpoint. So this checkpoint occurs during actual mitosis, near the end of metaphase. And what it's looking for here is, are the sister chromatids appropriately attached to the spindle fibers? And the reason why it's going to be looking for that attachment is that's going to determine, will those chromosomes, will those sister chromatids separate, and will those chromosomes separate properly? So if they're not attached properly, you could end up with some of those mutations that we talked about last year, like having a trisomy or a monosomy, right? Where one cell is going to end up with two copies, and one cell ends up with no copies. Okay, so those are pretty grave mutations to have chromosomes completely missing. So it stops, it checks, it makes sure everything is hooked up properly, the spindle fibers are hooked up properly to the sister chromatids before it starts yanking them apart. So what happens if the cell screws up? What happens if the cell doesn't pay attention to the signals? It doesn't listen to the internal or external information appropriately. if it pushes past a checkpoint that it shouldn't have gone through. What will happen essentially is out of control cell division. And out of control cell division is essentially cancer, right? So that's where cancer cells are going to come from, right? Our main symptom of cancer is a tumor or a mass because those cells were growing and dividing when they shouldn't have been, because cancer type cells aren't responding to certain cues from the environment. Some of those cues being density dependent inhibition. Basically, when it gets too crowded, the cells stop dividing. They're too crowded. They know that they're now starting to encroach on other cells' space, on other cells' nutrients, on the resources available. Okay, we watched the video on quorum sensing with the bacteria, right? Your cells are doing the same thing. They're talking to each other and they're realizing that it's getting too crowded and maybe they need to stop doing cell division for a little bit. Anchorage dependence as well. Okay? Most of your animal cells need to be anchored into something. Okay? They need to be anchored into some underlying tissue to be able to actually divide. So this goes hand in hand with the overcrowding. If there's not enough space for them all to anchor in, then they will stop dividing. And this is one of those things that cancer cells could ignore. Cancer cells ignore these density dependent inhibition signals. They ignore these anchorage signals. Okay? They also ignore checkpoints sometimes. And so now I get lots of cells growing and reproducing when they shouldn't. That may also cause cells to go through apoptosis. Remember, apoptosis is our fancy term for cell suicide. Okay? And the cell has basically had a programmed cell death. And that could be because it is not being regulated properly. And so hopefully we'll be able to do some work in class, maybe talk a little bit about like tumor suppressor genes and kind of all the things that need to go along with really kind of all the things that need to happen to turn a cell into a cancer cell, for a cell to start, or the things we know about that need to happen for a cell to turn into a cancer cell, for it to start ignoring these cues that it's getting. Okay, but for now, okay, we need to understand that that's what's happening with these cells. They're ignoring these inhibitory signals that they're getting from the environment, from the surrounding cells, and therefore they're continuing to divide when they should not. Okay, so we'll spend a couple of days in class working on both cell cycle, looking at cancer regulation parts of this.